The title of our article is Loss of SMAD4 Promotes Colorectal Cancer Progression by Recruiting Tumor-Associated Neutrophils via CXCL18 CXCR2 axis. It was published in 2019, and below we have listed the authors. So to better understand our article, some context on the SMAD family along with TGF-beta might be needed. So TGF-beta stands for Transformation Growth Factor Beta, which is a little misleading because this cell signaling pathway is going to inhibit growth in most cell types. And for that reason, it tracks that TGF-beta receptors, along with the SMAD family involved in this pathway, are commonly mutated or deleted in many cancers. Instead of promoting growth, it will promote tissue maturation and stabilization. It can do this by facilitating in synthesis of ECM proteins, that is extracellular matrix, as well as promoting synthesis of inhibitors of serine proteases, which under normal circumstances can go on to degrade the ECM. So we've included here a schematic of the signaling pathway, starting with TGF-beta, a ligand binding TGF-beta receptor. SMADs can be generalized as RSMADs, which stand for receptor dependent, co-SMADs, which will help with translocation of SMADs into the nucleus, and then iSMADs, which can serve to inhibit this pathway, and we see this legend here. It's common that two phosphorylated RSMADs will bind important as well as a co-SMAD like SMAD4 here, which is a critical piece of our journal article if you recall from the title. From here, movement into the nucleus where the SMADs can bind genes with TGF-beta responsive elements. And then also in this nucleus, there are going to be phosphatases that can come along and dephosphorylate these SMADs, so that way they can be trafficked back into the cytosol after binding exportin. Completion of this cell signaling event can prevent cell cycle progression. If you recall the following information from our text and from our lecture slides, SMAD and TGF-beta can promote P15 INK4B and P21 CYP1, which are both CDK inhibitors capable of preventing replication and division. So we can see here where this occurs. TGF-beta signaling can also inhibit MYC mRNA activity if we follow the pathway here.
Some further background information on colorectal cancer. Colorectal cancer results from various mutations involving genes encoding for APC, KRAS, P53, and importantly, SMAD4. It's seen that in 30 to 40% of colorectal cancer cases, there is a loss of SMAD4, and this is correlated with a poor prognosis. It's also shown in mice studies that a loss of SMAD4 leads to the recruitment of myeloid cells via a CCL9-CCR1 axis. This is indeed bad because, as we know, tumor-stroma interactions are very significant to tumor progression, tumorigenesis, and the growth of the tumor, and we see these in tumor-associated macrophages and tumor-associated neutrophils, as well as an increased number of TAMs and TANs leads to a worse prognosis for the cancer as well.
In their previous studies, they found that a loss of SMAD4 from colorectal cancer causes an accumulation of CCR1 plus or CCR1 positive immune cells to infiltrate the tumor and facilitate invasion and metastasis. However, they wanted to explore other ways the loss of SMAD4 could lead to immune infiltration, as they hypothesize other pathways can play a role. In this study, they focus on CD66B plus neutrophils and eventually the CXCL1-8 CXCR2 axis. To investigate the relationship, they broke down their hypothesis into a series of questions and experiment, which makes up the rest of this paper. First, they were looking for a relationship between SMAD4 expression and neutrophil presence by looking for CD66B, a neutrophil marker. Figure 1a shows via H&E staining and IHC method the presence of those neutrophils in 43 SMAD4 positive and 56 SMAD4 negative colorectal tissues. IHC stands for immunohistochemistry. It's a method that uses antigen and antibody reactions to detect biomarkers. So the H&E staining is basically a control. It's to help look at all the tissue types just naturally without looking for a specific biomarker. Then we're looking for SMAD4. As you can see in the positive, you'll see like brown marks. Those brown stains are SMAD4. While in the negative cases, you'll see no brown, just blue, because there is no SMAD4. So, brown staining in the positive, no brown staining in the negative. Then when we look over here to CD66B staining, we'll see some staining here, some brown marks in the positive, but we see a lot more brown staining and therefore a lot more presence of these neutrophils in the negative SMAD4 tissues. So we're seeing this relationship visually between a loss of SMAD4 and the increased presence of CD66B. And we'll actually, if we look at figure 1b, it's going to quantify that difference we saw in figure 1a. So, there it is. The SMAD4 negative tissue is going to have a higher and a significantly higher number of CD66B positive neutrophils compared to the SMAD4 positive tissue. So we're painting this picture. But before we move on, they wanted to look at overall survival. As you can see here, they further broke down the positive and negative SMAD4 groups based on the number of neutrophils present. So group 1 is positive for SMAD4 and have a high amount of neutrophil evasion. Group 2 is positive with a low amount. Group 3 is negative with a high amount. And group 4 is negative with a low amount. And in case you were wondering what is considered having a low amount of neutrophils invading, it's based off of this number, about 36. So if you have less than 36, it's a low number. And that was because it was the median amount of neutrophils present in the overall samples. So group 3 has the overall lowest survival rate. And if you were to compare group 3 and group 4, the only difference is the amount of neutrophils present. So the driving force for the low survivability is likely due to the invasions of neutrophils. So this is just a summary slide for figure 1. And overall, we see that a loss of SMAD4 correlates with an increase of CD66 positive neutrophils, which negatively impacts mortality. Next, I wanted to investigate how SMAD4 was leading to this infiltration. And the rest of the experiment, they'll be using 4 different colorectal cancer lines, which are right here. So you got SW837 and Calco2. As you can see from this Western blot, they are positive for SMAD4, while the other two, SW480 and HT29 or 29, is negative for SMAD4. However, for all of these lines, they are expressing the rest of the SMAD family. and the ACTB is just the control. Another thing they'll be using for the rest of the experiment would be a SMAD4 knockout. So figure B shows their results for creating that SMAD4 knockout out of the two positive SMAD4 lines. And they used they're trying out two different techniques. One is the short hairpin loops, which is shorthanded SH SMAD4, and the other is a small interfering RNA segment, which is how they're labeling it here, SI SMAD4. So the 1 and the 2 stands for two different methods or types, so they're testing out two different short hair loops and two different small interfering RNA segments. And as you can probably see, they're not able to fully knock out the CALCO2 lines with the small interfering RNA segments. And there's a little bit of expression here for their short hairpin loop here for number 1, but their second short hairpin loop for the SW837 line was pretty successful. And as you'll see in the other slides, they tend to use this knockout a lot, and it's because out of the other knockouts, it was the most successful. The other things here, ACTB and the scramble and the NT, they're all just controls. So now we have our lines, our baseline for us to work with for the rest of the experiment. We can now start looking for what is responsible for infiltration, and we're going to do that using a chemokine array. A chemokine array is just a type of amino assay. Once again, we're using that antibody-antigen reaction for detection, but instead of using tissues as a backdrop like we did in IHC, we're going to use a plate or a membrane. And now that we have our SW837 cells with that stable SMAD4 knockdown, we can compare the expression of these chemokines, which is just a type of cytokine, to the SW837 cells, which are SMAD4 positive. And we see that a major difference between the SW837 cells and the knockout cells was an upregulation of CXCL1 and 8. So we can see that right here. In the SMAD4 positive cells, we're not seeing as much of an expression. And you can definitely see the CXCL8 has a major increase of expression, while this one is a little faint, so it's a little harder to see. But if we go to the next two panels, they actually go ahead and quantify it for us. So using reverse PCR, they were able to see that once they knocked out SMAD4 expression in their two SMAD4 positive cell lines, the CXCL1 and the CXCL8 mRNA expression increases significantly. So then they move on to their next question. Is the loss of SMAD4 responsible for the different levels of chemokines? To answer that question, they introduce exogenous SMAD4 into the cell lines through a virus and to see the impact of expression. So figure F is a verification that the SMAD4 was introduced into the HT29, and SW480 cell lines. Specifically, they're checking the expression of their labeled exogenous SMAD4, which is this SMAD4. So that is their exogenous SMAD that they're adding in to the 2 cell lines. And you can see in G and H that when they introduce this SMAD into the cells, there's going to be a significant decrease in both the CXCL1 and 8 expressions. So if you look here, for example, you'll see in the controls, they've got normal expression, and then here, when they add that exogenous SMAD, they're like, oh, it go down. And it's a significant decrease. So that sums up figure 2. They did a lot, but it all boils down to essentially creating a knockout and then using that knockout to discover that SMAD4 has an inverse relationship to CXCL1 and 8. And then we're going to continue our investigation through figure 3.
So now that we've established that there is a connection between CXCL1, CXCL8, and SMAD4, the scientists wanted to figure out exactly how SMAD4 is regulating the levels of these proteins. So we know that in TGF-beta signaling, the ligand binding to our TGF-beta receptor leads to SMAD2/3 being phosphorylated and forming a complex with SMAD4. This complex is then able to translocate to the nucleus and regulate the transcription of genes. In our SW837 SMAD4 positive cells, TGF-beta does indeed lead to a decrease in CXCL1 and CXCL8. To investigate exactly what is going on and how they are being regulated, the promoter for CXCL1 and CXCL8 was transfected into these SW837 cells. The levels of CXCL1 and CXCL8 were then measured in the presence and absence of TGF-beta. The results showed that TGF-beta signaling had no effect on CXCL1 and CXCL8 activity. To further check this, a chromatin immunoprecipitation using an anti-SMAD4 antibody showed that there were no changes or alterations done to the promoters of CXCL1 and CXCL8, which goes to show that SMAD4 was not binding to or regulating transcription through the promoters of these proteins, these genes. So to identify how CXCL1 and CXCL8 were being regulated, SW837 SMAD4 negative cells were treated with kinase inhibitors. An mRNA analysis revealed that I-kappa beta kinase 2 inhibitors and indirubin were the prominent inhibitors of CXCL1 and CXCL8, meaning SMAD4 was regulating CXCL1 and CXCL8 indirectly through some other pathway. Now we need to investigate how these colorectal cancer cells are attracting neutrophils with CXCL1 and CXCL8. So we know that CXCL1 and CXCL8 are able to attract neutrophils because neutrophils contain the cognate receptor for them, CXCR2. It was theorized that the neutrophils were being recruited in SMAD4 negative cancer cells through a CXCL8-CXCR2 axis pathway. To test this, neutrophils were collected from healthy donors, and a flow cytometry was used to ensure that they were both CD66B positive and CXCR2 positive, shown here in figure 3A. The dotted lines show our isotype control, and our solid lines show our anti-CD66B and anti-CXCR2. Next, an in vitro chemotaxis assay was performed between the supernatant of colorectal cancer cells and our collected neutrophils. The isolated neutrophils were added to the upper chamber, and the colorectal cancer cells were added to the bottom chamber. The migratory response of neutrophils to the colorectal cancer cells in SMAD4 negative and SMAD4 positive control cells was then measured and recorded in a chemotactic index as seen in figures 3B, 3C, and 3D. In figure 3B, we see that both of our SMAD4 negative cells, shown here number 1 and number 2, had almost double the neutrophil migration than our SMAD4 positive control cells. Next, the effect of anti-CXCL1 and anti-CXCL8 antibodies were studied. Upon adding these antibodies, we see a suppression of neutrophil migration, with anti-CXCL8 suppression being much more prominent. So if we look at figure 3C, we once again have our control that is SMAD4 positive and contains no anti-CXCL1 and no anti-CXCL8, where it has a low chemotactic index indicating low neutrophil migration. In bar number 2 for our chemotactic index, we see once again our SMAD4 negative cell with no anti-CXCL1 or anti-CXCL8, and we see a strong chemotactic index indicating high neutrophil migration. Upon adding anti-CXCL1, we see that the neutrophil migration is cut almost in half, and adding only CXCL8 actually significantly reduces neutrophil migration to levels even lower than our control SMAD4 positive. And upon adding both anti-CXCL1 and anti-CXCL8 to the SMAD4 negative cell, we see a chemotactic index that is relatively similar to control levels. Lastly, the researchers wanted to study the effects of a CXCR2 inhibitor or the receptor inhibitor and its effects on neutrophil migration. They added SB2250002, which is the CXCR2 inhibitor, and then measured the levels of migration. We once again have both of our controls that are SMAD4 positive, and then we have our SMAD4 negative cells, in which we see that no receptor inhibition has strong neutrophil migration, but upon inhibiting our receptor, we see almost no neutrophil migration. The level is practically zero. So these results actually show that the neutrophils are being recruited to our SMAD4 negative colorectal cancer cells via a CXCL8 CXCR2 signaling pathway.
We got to see a little bit of immunohistochemistry in Nadine's explanation of figure 1A. Here we again use immunohistochemistry methods, but for panel A of figure 5. So we'll walk through a couple of key takeaways. First off, CXCL1 and CXCL8 are both abundantly expressed within tumor-associated neutrophils in SMAD4 negative colorectal cancer samples over their counterparts being SMAD4 positive colorectal cancer samples. It's worth noting that we do see more staining for CXCL1-8 in the positive over normal. However, SMAD4 negative is really what's going to take the prize here, and we can see this staining quite clearly going in a progression from normal to positive and to that quote-unquote abundant expression with SMAD4 negative. And right here is just a great callback to the title. If you recall, the way in which SMAD4 loss recruits tumor-associated neutrophils via CXL1 and 8, and then we can make that intuitive statement that CXL1-8 expression is inversely proportional with SMAD4 expression. It's also worth mentioning that myeloperoxidase, MPO, is a human myeloid lineage marker, and why that's important is because that was included here because when we move on to the immunofluorescence staining, the anti-CD66B antibody, which we know to be characteristic of granulocytes, a family of which neutrophils are a part of, that didn't give a signal. So this poses no conflict, as we see here that the distribution between MPO positive cells was almost the same as the CD66 positive cells. Maybe we see a little bit of a difference, but the authors of the journal article say that this is not much. Panels B and C focus on a simultaneous immunofluorescence staining. In panel B, we can see that there is a simultaneous immunofluorescence staining for MPO, which is in red, CXCL18, which is in green, and then the DAPI is blue. The DAPI is significantly yellow in CXCL1, but more so in CXCL8. And then the majority of cells expressing CXCL1 and 8 were positive for MPO, which suggests that CXCL1-8 is secreted by tumor-associated neutrophils. Here we're switching over to panel C. So it's worth noting that pro-tumorigenic tumor-associated neutrophils can be characterized by their angiogenic function by producing the matrix metalloproteinases, which we know already a great deal about, abbreviated MMPs, as well as immunosuppressive function by producing ARG1 and IDO. Consistently, they were able to show and support that the majority of these MPO positive cells were in fact CXCR2 positive and expressed MMP2, so that would be matrix metalloproteinase 2, in addition to ARG1 and IDO. Within this immunofluorescence staining, there is staining of CXCR2 in red. Then there's several stainings present for MPO, MMP2, MMP9, ARG1, as well as IDO, which we see in green. And then the DAPI stain is, of course, blue in these tumor cells. The DAPI is blue in MPO, IDO, and ARG1, but yellow in MMP2 and MMP9. Lastly, the majority of MPO positive cells were CXC2 positive and expressed MMP2, MMP9, in addition to ARG1 and IDO.
So I will be talking about figure 6, and this entire figure signifies about the clinical significance of CXCL1 and CXCL8 in colorectal cancer. So in figure A, or panel A, we are looking at more of like a dot plot in which we are looking at the relative mRNA expression in neutrophils in your tumor over your blood. So your CXCL1 and your CXCL8 are your neutrophils, and this entire dot plot is using qRT-PCR, which is a real-time PCR that monitors the amplification of a targeted DNA molecule during the PCR. So the average in CXCL1 is 4.7 tumor over blood, which shows that that's like a very small and bunched up expression compared to your CXCL8 because it has a wider range, and the CXCL8 expression or concentration or mRNA expression is your 38.8 tumor over blood, which shows that CXCL8 mRNA expression is relatively higher than compared to CXCL1. So then what we're going to do is on the second panel, what's going to be tested is the CXCL1 and CXCL8 in concentration in both your control group, which is your healthy donors, and your patients with colorectal cancer. So in CXCL1, if you see in your healthy donors, you see that there's a high range of mRNA, not mRNA, but CXCL1 concentration, which is around 100.4 picograms per milliliter. But compared to your patients with the colorectal cancer, there's only 8.8 picograms per milliliter, showing you that your CXCL1 concentration in your patients with colorectal cancer is a very low compared to your healthy control group or your healthy donors. However, in CXCL8 concentration in your colorectal cancer is much more higher compared to how the outcome is in CXCL1, and you can see there's like a higher range, and that the average is around 13.7 picograms per milliliter. However, in your control group, in your healthy donors, it's very low and bunched up, and it's around 2.9 picograms per milliliter. This already shows that like how important CXCL8 is in our colorectal cancer cells. So the whole point of panel C is to see the CXCL8 concentration in each stage of colorectal cancer. So in your control group or your healthy donors, you see that your CXCL8 concentration is very low. It's at 2.9 picograms per milliliter, and it's slowly starting to increase at each stage of cancer. So here you have a 5.4 picograms per milliliter at stage 0 to 1. Then you have 20.3 picograms per milliliter at stage 2, and then it's like a little bit higher. Your stage 3 has the highest, which is 20.8 picograms per milliliter. However, in stage 4, you have a very almost low in the median range of CXCL8 concentration. It drops from 3 to 4. And this is because that increase and decrease is because this is all reflecting the tumor size. So in stage 2, your tumor size, or the median tumor size diameter, was 45 millimeters. And that was the same for stage 3. That's why you see a little bit of an increase. However, in stage 4, since your cancer is at the point of metastasizing and it's moving at other areas, your tumor size is a lot more smaller. And in this case, your tumor size is around 40 millimeters. And that is why there's that sharp drop from stage 3 to stage 4 of colorectal cancer. Now, after that, we're looking at the clinical significance of CXCL1 and CXCL8 in colorectal cancer. And this is panel D, and it's looking at both of the overall and relapse survival of both CXCL1 and CXCL8 patients. To find this p-value, they do a log rank test. This entire graph is based on a log rank test. And based on the high p-values, which is over 0.05, you see that these results are not statistically significant. And if you're looking at CXCL1, there's no association between the CXCL1 concentration and prognosis basis on the high p-values. However, in CXCL8, you see a much more, almost close to the p 0.05, and it shows that the high CXCL8 levels has a shorter overall survival and relapse survival compared to the low ones. And although it is not a statistical difference based on the high p-values, or somewhat high p-values in this case, this shows that CXCL8 couldn't be a novel biomarker of colorectal cancer. However, it is associated with poor prognosis. In this panel, which is panel E, we're going to look at a better outlook and diagram of the cells involved with colorectal cancer, and basically how angiogenesis is induced. So as we talked about earlier, angiogenesis is one of our hallmarks of cancer, and in the past exam, we had discussed that angiogenesis is basically it's forming new capillary growth based on pre-existing blood vessels. So the cells involved is your CXCR2+, or your neutrophils, right? Your neutrophils so you have your neutrophils, and your CXCR2 is bounded to your neutrophils, and it's just going on its way in its blood vessel. That's what's normally happening. And then you have your CRC, your colorectal cancer cells, outside of your blood vessel. But then when you lose this SMAD4, this causes your colorectal cancer cells to increase their expression of CXCL8 and CXCL1. So this causes your CXCR2 and your neutrophils to get recruited to these cancer cells. And this causes the expression and secretion of CXCL1 and CXCL8 to even increase even more, to speed up the process of neutrophils to come in by the cancer cells and promote this tumor progression, and eventually promote the angiogenesis. And this is due to releasing the MMP2 and MMP9, which are both matrix metalloproteinases that degrade the ECM to recruit these extra fibroblasts and endothelial cells to promote angiogenesis and for it to move on more. And it also releases your ARG and IDO as well. And this kind of causes the vessels to form until your tumor is done. It's at this point. Overall, on this figure 6, I've came with a lot of discussion and conclusion. Your SMAD4 contributes to your myeloid cell infiltration and your tumor progression. And it also regulates your CXCL1 and CXCL8 expression. And this is only done with the IXB kinase 2 and GSK3 recycling-dependent kinases. This shows, based on the figure 6 that I showed you, panel E, it shows you how important SMAD4 is, because without SMAD4, these colorectal cancer cells, these tumor cells will go crazy and try to express the CXCL8 and CXCL1 even more and more and more. CXCL1 and CXCL8 are both pro-inflammatory mediators, and they have a relation to your stromal cells because they are secreted by a variety of neutrophils, monocytes, fibroblasts, and endothelial cells. They're also found in your chromosome 4, and they're modulated by promoter methylation. CXCL8 is shown to be a potent androgenic molecule. As you can see from the figures that we have discussed previously, how it's so important, especially in so many colorectal cancer cells, like how high that expression is compared to your CXCL1. Also, the loss of SMAD4 in your colorectal cancer cells, as I've discussed, it results in your CXCL1 and CXCL8 to recruit the neutrophils, which is another one, which is CD66B plus by your CXCR2. And this basically is just recruiting the neutrophils that produce so much CXCL1 and 8 to accumulate more CD66B plus neutrophils. And this just amplifies the cytokine and chemokine milieu within the tumor microenvironment. CXCL8 is shown to be a potent angiogenic molecule due to its inflammatory cytokines, its chemical environmental stressors, its pathogens, and like its reaction by oxidative stress. So like looking at how CXCL8 and how it is to these cells is surprising and shocking to just see like how important SMAD4 is within the relation of your myeloid cells, your colorectal cells, your neutrophils.
We agree with the conclusions they've made. The research they have done is very extensive and thorough. However, they can go farther with this work. For instance, since a loss of SMAD4 correlates with a significant decrease in overall survival, and CXCL1 and 8 promote major hallmarks of cancer, why does it not also correlate with a significant lower survival and relapse? What other factors are we not considering? Also, remember that this work was inspired by their previous findings regarding SMAD4 and CCR1 plus immune cells. What other ways can SMAD4 impact the tumor microenvironment? And finally, they have concluded that this was a good biomarker. How can we use this information for treatment?